So, this lesson is going to be on mutations and their effect on protein synthesis. So, just to kind of get you thinking along the lines of mutations, if you've ever seen the X-Men movies, um, if you remember Cyclops, he was actually born with his mutation. Most of the X-Men were. But if you look at like Hulk and Spider-Man, they were actually acquired their mutations. So Hulk was exposed to radiation, and then Spider-Man was bitten by the radioactive spider. In both of those cases, though, those there were mutations to their DNA um, that caused different characteristics than would say if they didn't have that exposure. So during this lesson, we'll talk about some different types of mutations. We'll mention a little bit about environmental factors that can influence those, and then also talk about their effects on protein synthesis. So we're going to come back to the silent mutations, but um, as you're filling out the chart, you don't necessarily have to draw pictures unless you want to. As long as you kind of get the general idea of it, you don't have to write every single bullet point in the notes if you don't think that would help you. So, the first types of mutation we're going to talk about are small scale mutations. These are mutations that just affect the actual DNA itself, like the base pairs or the sequence of base pairs. It occurs during replication. It can either occur in replication in meiosis, like in your body cells, or I'm sorry, meiosis in your sex cells, and then that mutation would then be passed on to your offspring, or it can result just in mitosis in your body cell replication. And in that case, remember, it would not be passed on. It's just a mutation that occurs in your body only. So, in our DNA sequence, this is kind of skips a few steps here, there, in the center, but we have our DNA, and if once we've translated this to RNA, you see they've kind of already split it up into our codons. So when we read RNA, we read them in three base pair segments, and we call those codons. So if I took this DNA and if I transcribed it into RNA, and then I translated it, these would be my chain of amino acids that would then eventually form a protein. So if I take one of those nucleotides and I replace it with something else, like for example, let's flip back. You can see this was CCT, now it's CGT. So I've just replaced one single nucleotide. We call that a point mutation because it only happens at one point. Sometimes it's called substitution, so you may see that as well. But like I said, it just occurs when um, one nucleotide is replaced. And then what happens is you end up with a new amino acid sometimes. Okay, so in this case, it codes for a different amino acid. Another type of small scale mutation is a deletion. So in this case, we deleted a, um, a nucleotide or a base pair or a one base. And then what happens is it actually causes what we call a frame shift mutation. So it kind of shifts our entire reading frame, right? Because we read in threes. So it shifts everything down one. And because of that, what it's going to do is affect every single amino acid from the moment that mutation occurs. So it's almost like a chain reaction. Once I shift one letter over, I've messed up how I'm reading everything in threes. Another frame shift mutation is called an insertion. It's just like what you would think. We've inserted an additional nucleotide. And again, that causes you to read different sets of three. So it's like, you put um, if I have groups of three and I try to put one in the middle, I have to regroup everything after that to remake my groups of threes. Um, our large scale mutations, we've already talked about these a little bit when we talked about chromosome mutations in the last unit. That's what these are, but I feel like it's important to mention them again here as well, just so you can kind of compare them to the small scale mutations that occur with the DNA sequence. So, a large scale mutation, it's just a mutation that affects the entire portion of the chromosome. Um, so it's we're not looking at just a single point or a single moment in the DNA strand. We're looking at an entire chromosome, and in this case, entire genes and sets of genes are completely altered. So we're not, changing just one sequence, just one little gene here and there. We're changing multiple genes, so it's a pretty big deal. So we talked about a deletion. So here's just a picture of that. This is where one whole area of the chromosome is lost. So you can see how it's shortened now because we've kind of lost that middle section. For a duplication, it's just like what it says. We've duplicated a section. And so in this case, now you have extra genes that were already present. And so this can cause, you might think, oh, that's not a big deal. It's just extra, but it can actually cause long term effects down the road. Inversion, this is just where you can kind of see from the picture if you look at the segment here, this wider segment. All we've done is flip flop it. So we've kind of flipped it upside down and stuck it back in. Um, this means that now um, your gene is there, but the actual way that we're reading the gene, right? Because we read from your your transcription occurs beginning at the three prime end and it moves towards the five prime end. In this case, we've kind of swapped the section. So now, where I used to read 3 to 5, now it's going to read 5 to 3. And so, where we would make a complementary strand, that's also going to be inverted or backwards. And so, it's gonna alter eventually alter our protein synthesis. Insertion. This is where we've taken part of one chromosome and inserted it into another. Um, this Y It depends on what happens and where it happens and what type of result you're gonna see. You may not notice, like, if this happened in my, um if this happened during, the meiosis process of the egg that I was fertilized with, I may not notice a difference because I still technically have all of the parts needed for, transcribing and translating these genes. However, when my cells go to divide during meiosis to produce my sex cells, um, if there's a chance that the offspring or my future child would be missing or have extra of something depending on which one of these chromosomes they got. So if they ended up with this chromosome 20 that has part of chromosome 4, now they're going to have extra parts of chromosome 4 because they're already going to have a chromosome 4 from the father, and now all of a sudden they have extra chromosome 4, or maybe they have um, um, a different copy of my chromosome 4. So you end up with more issues further down the generation line. Translocation, this is similar to um, an insertion, but they're swapping in this case. And so again, this is one of those that you may not see very too many issues right away, just depending on where exactly those they were swapped. But down the line, once I have children, I might start seeing some issues because you can end up with extra copies of chromosomes or missing parts of chromosomes. We talked about non-disjunction. This isn't necessarily a mutation. This is more of just a error in meiosis, or where it's not you're not getting the chromosomes that you need. So this is where you have either an extra chromosome or a missing chromosome. So technically, this isn't exactly a mutation, but we're just kind of lumping it in with those other chromosome disorders, just so you can kind of see some of the differences between the small scale and large scale. And so this is where we're going to kind of go back up to the top of the page where it talks about effects of mutations. So sometimes a mutation can have no effect at all, and sometimes it might lead to incompatibility with life. So it just kind of depends on what happens or what the mutation is. So the first one is a silent mutation. And this is like if I had a point mutation where I just changed one letter, if it's still coded for the same amino acid, then we would call this a silent mutation. I would never even know. You may never even know that you had this mutation because nothing has changed. You're still producing the same protein. A missense mutation means that we are going to notice something because instead of you have a some type of change, whether it be a frame shift mutation or maybe it's a point mutation that leads to a different protein being produced. So you do have a different amino acid and a different protein that's produced, which can change the function um, of that protein. Nonsense mutation is where your codon, where whatever your new codon is after whatever mutation occurred, results in a stop. So that might mean that your gene isn't fully transcribed, or I mean, or translated, because instead of continuing let's say there were 20 codons that needed to be translated. Well, if the third one codes for a stop, all of a sudden you have this shortened protein, so it's not going to function correctly. Large scale mutation effects are much larger. This is kind of like what we had talked about last unit, where these large segments of chromosomes could be missing, or you could have extra. So you can end up with, trisomies, or you could end up with this is kind of where we see more like a miscarriages that occur during the first trimester are usually because of something like this, some type of chromosomal mutation where you have this large scale error, and it doesn't necessarily isn't necessarily compatible with life. Um, so this is what I said. Often large scale mutations lead to cells that aren't viable. They're not going to live. Um, the last thing is a lot of times these mutations occur because you've been exposed to some type of chemical. So after what we're going to kind of talk about um, towards the end of the semester when we kind of start getting into our cancer unit, we'll talk about a lot of times really the reason that the older you get, the higher your chance of cancer is because your body's just been exposed to so many chemicals throughout its life, and radiation and just things that your these mutations just kind of slowly start to build up, and you end up with a mutation in, a tumor suppressing gene or an oncogene, and so it ends up that the end result is this unchecked growth, which leads to cancer. This is just an interesting application that you don't necessarily have to write it down or anything, but um, humans, we've been intentionally mutating plants and things, animals for thousands of years. Um, even before we really knew what we were doing, farmers have been mixing, plants and types of plants and types of fruit for years, not really necessarily understanding what they were doing, but that's what was happening. Really, are these there were mutations that were occurring that would result in, let's say, a larger watermelon that has fewer seeds. Um, breeding chickens that have more white meat or, or larger, things like that. So there's actually been more um, of this going on than even before I really intentionally knew we were doing it. Um, Now that we can intentionally manipulate the genetic code, there's lots of things that are done that maybe aren't always up to code, maybe a little bit controversial. So for example, like disease resistant papaya. So the fruit doesn't rot because you've genetically altered it. Drought tolerant corn. Um, there's been, obviously, this is a super, super controversial one, but possibly editing genes while a child is developing, which could maybe prevent a child from having a certain disease later on in life. So, if you know that they're going to be at risk for something because they have a certain gene, can you edit that gene while they're developing to prevent that later on? We're going to skip that. So, that is it. Um, so, just make sure you know the basic definition of the mutation, and kind of what the end result is. And then, like I said, the second thing you're going to do today is a little worksheet that you'll actually practice identifying the type of mutation, and creating that mutation, and then kind of seeing that end result.